This is a GMO. And this is a COVID-19 swab test. What are their similarities? Well, they both involve genes. This tiny segment of DNA contains information that codes for protein that is expressed as a trait. Traits that can be inherited from your parents. For example, your eye color, your skin color, the texture of your hair, your blood type, and so much more. So basically, genes are the one who would tell our body what we would look like. So how do scientists find out all this information from just a small molecule that even our eyes can see? How do scientists know if you have the virus for COVID-19? How do they manipulate genes to make GMOs? The answer is polymerase chain reaction. This tiny machine paved the way for many discoveries. It can make millions of copies of genes in just a few hours. So how does this work? Simple, by using the principle of DNA replication. In polymerase chain reaction, or PCR, first they take the DNA of the target organism where all the information is contained, including the genes. Once this is isolated, the scientists use cocktail of enzymes that start the replication through cycles of heating and cooling. Primarily, this cocktails contains primers and polymerase. These are short pieces of single-stranded DNA that researchers designed to only bind to a specific target DNA sequence. Polymerase catalyzes the synthesis and replication of new DNA strands. There are 3 steps in PCR amplification: denaturation, annealing, and elongation. The isolated DNA serves as the template for denaturation. As temperature is increased, the double-stranded DNA is opened up, making separate single strands of DNA. The second step is primer annealing. Here, the temperature is lowered. The primers bind and read the target sequence of the DNA, like a gene, in opposite directions. Now, in the third step, is the elongation of new DNA strands. Temperature is raised again for the elongation of the polymerase, creating new double-stranded DNA strands. Each of the new double-stranded DNA strand consists of one old strand from the original template and one new strand created as the template's reverse complement. Now we call this semi-conservative replication of the sequence. And that's it. That is how PCR is done. PCR is useful in genetic engineering. It might not be noticeable, but in our everyday lives, we can encounter things that undergo PCR.